Hello students, welcome back to my channel CSIR Buddy, and this is the fourth part of prokaryotic replication, and that is termination. In the previous lectures we have already studied about all the things that is related to replication, initiation, elongation we have already completed. I will be providing the link of the same in the description box. So if you haven't watched those videos, first go and watch those videos, and then only you will be able to understand this one lecture. So termination, what is termination? When the DNA replication is completed, we have got two strands. So we are talking about circular DNA replication, and now we have already got two strands out of it. Now we need to terminate this replication, that is from the URI side. Now we have to terminate this replication. Termination of replication takes place at the termination site in the prokaryotic DNA. DNA replication terminates when replication forks reach specific termination sites. So there are termination sites which are present in the replication fork that are known as TER sites. That is, replication forks meets each other on the opposite end of the parental circular DNA. So that means they meet each other in a opposite direction, because the replication is going on up in an opposite direction. The two replication forks are synchronized by 10 to 23 base pair TER sequences. So TER sequences are present that bind TUS proteins. So TER sequences bind with TUS protein, and TUS is a termination utilization substrate. TUS proteins can only be displaced by replisomes coming from one direction. So what are replisomes? All the primer plus helicase. So these are replisomes. So TUS proteins can only be displaced by replisomes coming from one direction. So TUS proteins could be replaced by replisomes which were present in the DNA sequence. TUS protein binds to terminator sequences that are TER sequence and acts as a counter helix when it comes in contact with an advancing helicase. The bond TUS protein effectively halts DNA polymerase movements. So TUS protein will halt or inhibit the DNA polymerase movement, and this DNA polymerase will be not able to make further DNA replication. So what was DNA polymerase? DNA polymerase was an enzyme which was helping to add on new nucleotides to the DNA strand. So now it won't be able to do that. So I think this theory or this summary is clear to you all. As you can see in this figure that we have origin of replication and the replication fork, and here the replication is stopping. So what is happening? That the replication is going bidirectionally. The replication is going bidirectionally. While these strands are going opposite to TER sequences, that is TER C, B, F, G, A, TER D, TER E. So these sequences are moving upwards, and this one replication of DNA is going in a downward direction. If they are opposite direction, they will be allowed to move very easily. They can pass very easily. But when they come into a same direction, here the replication stops. Same goes for this. When this one strand is passing on this one strand, it will let it go. That is, TER sequences will let it go. As this one fork starts coming from the upward direction to these TER sequences, then here also replication would stop. So TER sequence would not allow the further replication to happen. The TER, termination, and TUS, termination utilization substrate, TUS is a protein, and TER make a TUS-TER protein complex, and this is how the replication stops. So now supercoiling. What happens is when there are suppose two strands, and when we try to separate these strands and make a bubble, then there are some supercoils. Like you can have a example of a thread. If we take a thread और दोनों thread को we try to pull it apart, then what will happen? It will make some of the bubbles, or we can say that supercoils will be there. So in order to remove these supercoiling, we need an enzyme that is known as topoisomerase. So topoisomerase is an enzyme which release the positive supercoiling. So these supercoils are known as positive supercoiling. Supercoiling is of two types, positive and negative, and we are talking about positive supercoiling. So topoisomerase is also of two types, type 1 and type 2. Type 1 break one strand only. So it makes a break in a single strand. So if this is a DNA molecule, so it will make a break in a single strand. And there is no need of ATP. There is no requirement of ATP, and the linking number for this is number 1 or the multiples of 1, that is 1n. Type 2 break two strands and it requires ATP, and linking number is 2. One more thing to note is that type 1 is a monomer. It has only one unit, while type 2 is a heterodimer. It consists of two subunits. So we will further study about topoisomerase. DNA gyrase. Type 2 topoisomerase is a heterotetramer, and it is made up of gyrase A and gyrase B. So gyrase A is responsible for the breaking down of the phosphodiester bond. So in this case, what will happen if there is a supercoil somewhere over here? It will first break down this bond and then attach these two strands in a proper manner and will release the supercoiling. So in case of type 2 topoisomerase, the gyrase B is helping in ATP activity. That is, it is providing an ATP energy to break down this bond. So gyrase A is responsible for breaking down the bond, and gyrase B is responsible for the ATP energy which is required. Now inhibitors of topoisomerase. These are the inhibitors of topoisomerase which you have to remember. These have been asked in previous year questions in CSIR net. Quinolone antibiotic, nalidixic acid, norfloxacin, and gifloracin, novobiocin. So these are the inhibitors which you have to remember. And I hope that this lecture was clear to you all. If you like this lecture, so please like, share and subscribe to my channel. Thank you.